All right, now we're getting into one of our big ones, and that is vision, right? Vision and hearing are some of the big ones, vision especially. So we're going to do some recap of the anatomy of the eye. We're going to talk about how light passes through and some of the cavities involved in the eye. We'll get into the lens and the cornea and how they help with the function of the eye, and then how the eye is under sympathetic and parasympathetic stimulation. And then we're really going to hit on a lot of things involved with the retina and how the rods and cones allow us to detect visual stimulation, right? Light stimulation. And then get into, the visual cycle, adaptation, color vision, as well as some disorders. So just as a quick review of the eye's anatomy, here's our picture of the eye. I'm just going to point out some pieces. I'm not going to point out everything necessarily, since this is the focus is on physio. But we do want to point out a couple things, namely the cornea, the pupil and the iris, the lens, the ciliary bodies, and the suspensory ligaments. And then back here we have the three layers of sclera, the choroid, and the retina. The retina is the one that we're really going to talk about, and the optic nerve here, right, as well as blood vessels that will service that, branching out throughout the eyeball to service the retina and whatnot. There are also muscles like the lateral rectus muscle, the medial rectus muscle that are used for controlling the eye's movements, right? Those are not going to be controlled by the optic nerve. We have a separate cranial nerve called the oculomotor nerve that will control those. Right? Again, if you need to pause here to take a second to look here, by all means, go for it. I named most of the things. Some things I haven't named yet, and we'll talk about them as we get to them, but do like a quick review so you understand the different parts. And this is just a table that can help you understand it a little bit better so that you can kind of take the time to understand the different parts. You may see things like the retina called the neural tunic, the vascular tunic or the choroid, and you may see like the sclera called like the fibrous tunic. Those again are just somewhat interchangeable terms. And then everything else here is just simple in terms of location, function, composition. So let's talk about how light passes through the eye, and we're going to simplify it relatively, and we're going to simplify it a little bit so that we're not going too crazy, because again, we're not worried about the anatomy so much, we're more worried about how things work. So light is going to enter in through the cornea, it's going to pass through the aqueous humor that we have here in the anterior chamber, through the pupil, right, through the lens, through the vitreous humor in the posterior chamber, and then the retina, primarily trying to hit on this fovea or the fovea centralis. Right? If we look closer, this is what we would see for that retina, right, going back. But this is the path that light is taking, right, through all of these components. So we're going to talk about at least a handful of these components and how they contribute to vision. So let's start with the lens. The lens is one of the big pieces that's going to help with this. In addition to the cornea, we're going to go a little bit out of order, so don't mind me with that. But the lens is going to be what refracts light, basically to try to appropriately focus light on the retina. The big thing we're trying to do is focus that light on one single point on the retina called the fovea centralis, or the fovea if you're looking at it shorthand. In that fovea, we see a high number of cones, which is used for visual acuity, or like that crispness of vision. The lens can move, it can change shape to maintain its ability to focus light on the fovea, and that change in shape is an autonomic nervous system reflex. So this lens can accommodate, meaning it can change shape to help you focus on something that's nearby or something that's distant, right? How we change, we'll talk about that in a little bit, but keep that in your head for now, that the lens is able to change shape. The things that are helping the lens change shape are the ciliary bodies, which are these tiny muscles, and the suspensory ligaments that are holding the lens in place. And so how those move is going to be what allows that lens to change shape for visual accommodation. Another key part is the retina. The retina is this inner layer of the eyeball. It's basically like a big screen for light to hit. And again, I mentioned it earlier, but I'm mentioning it again, the fovea centralis. Again, this is the area that we are trying to focus light on. All right, so the fovea centralis is kind of near the central point of the retina. The central point is also called the macula lutea. But the fovea centralis is the piece that we really care about because this is where we have a lot of cones, and cones are good for color vision as well as detailed vision, right? Getting that crispness in your vision. So the highest visual acuity, basically this is the point where your vision is going to be the clearest, and so this is why we want to focus light on this single point. There's near this though, a little bit to the side, right towards the nose, we have another spot called the optic disc, also called the blind spot. This is where your optic nerve and blood vessels connect to the eyeball, and because the optic nerve is there, we cannot have any rods or cones. And since there's no rods or cones, there's no visual input, right? And we can actually test for the blind spot to find it. We just have to basically, have a blank piece of paper, draw a dot on it, cover one eye, and then move that dot around towards your eye until it disappears. And when it disappears, that's when you found your blind spot. So if we take a look over here of why the blind spot is a blind spot, right? We see this optic nerve, it comes out and around. Well, right here, right in this area here, that's where all of those neurons, the axons and stuff, are traveling through here. And because of that, there are no receptors. There's no rods, there's no cones right here. No rods and cones means no visual input. No visual input means blind spot. That retina is also composed of several layers. We have blood vessels that are coming forward through the retina that are bundled together with the optic nerve. We saw them in between, and then they branch out across the retina because we need to service them, right? They need oxygen, they need all that kind of stuff, just like anything else. And then we have a couple layers. We have a pigment layer and a neural layer. The pigment layer is going to be right here. This is where we see the epithelial tissues or epithelial cells. They are going to basically absorb any extra light that never got picked up by a rod or cone. So they have pigment to just, kind of absorb that light. And again, this is at the very back. So right here, this is the very back of the retina is where we find that pigment epithelium. And then right in front of that, we see these, couple chains of neurons, couple chains of cells. This is the neural layer. This is where we have the photoreceptor neurons, like the rods and cones. We have bipolar neurons and then ganglion neurons that connect. So the bipolar neuron is kind of the go-between between the receptor and the ganglion neuron. If we look here, right, this is the actual picture of what it looks like. We see that pigment epithelium. See, it's really deep and dark right there. We have our rods and cones right here in this pink and purple mix. Then we have the bipolar cells spanning that, ganglion cells here, and then the optic nerve axons, which are going to travel through to the optic nerve and exit out the eyeball. And like I mentioned, we have two kinds of receptor cells, rods and cones. Rods are going to be responsible for your black and white vision, like grayscale, and night vision. So they work well when light levels are lower compared to cones. But they give you no color, right? It's just gray. Cones are for your color vision. They help give you a lot of detail, but they require more light, right? For them to actually function needs a higher level of light. So you probably notice that your color vision is not as strong as your night, as it's not as strong at night, right? At night you can generally see shades, right, of things, but color doesn't really stand out. So rods are better in lower light conditions, right? So it allows you to have good night vision and it gives you black and white vision, whereas cones, they need more light to function well, and they're involved in your color vision as well as that crisp, clear image you get. If we look at the pathways to the optic nerve, light is going to hit the rod or cone, then travel, we're going to send that impulse through the bipolar cell, then through the ganglion cell until we get to those axons so that we can travel out through the optic nerve to the brain. So what's this pathway look like, right? We kind of mentioned like, okay, here's some cells, here's some neurons that take us from the retina onward, but what's the actual pathway, right? When we take these rods and cones and we convert this light into a neural signal that travels through the optic nerve. So we get to the optic nerve, then what, right? From the optic nerve, we're going to travel through the optic chiasm, which is this crossover right here. So optic nerve, right, on the left and right, and then right here, right, is the optic chiasm where we kind of cross over. Then we have the optic tract, we pass through the thalamus, and then the optic radiation. See all these, we kind of like spread out, like we radiate, right? Spread out until we get back to the occipital lobe in the cerebrum, and we join up with the primary visual areas of the occipital cortex. Now, there's a couple things that we need in order for vision to actually work. We kind of went over the basics, but we're going to talk about some what I call visual processes. These are all things that need to happen in order for vision to be clear and you know, clear and pretty, right, and something we can actually use, right? So one thing that needs to happen is what's called refraction. Refraction happens anytime light passes through a transparent substance, whether it's water, air, whatever, right? When we pass through a substance with one density through a second substance with a different density. So you've probably seen this if you've ever looked through a glass of water, right? Everything is transparent, but as the light passes through the glass and through the water and through the glass again to your eyes, it refracts, it changes direction, right? It gets bent basically on its way to your eye as it travels through the glass. Your eye uses this to its own advantage, using the cornea and the lens, right? Light is coming from all different directions, and the cornea and the lens will refract light rays as they enter in an effort to focus light on the retina, so that if we look at this top picture here, this eye, light's coming in, we see that the cornea bends light in towards the lens, and then the lens further refines that, right, bending light towards the fovea centralis, the single point in the back of the eye. This is what's going to give us that really clear picture. Now, there are cases where someone's lens or cornea is not smooth in shape. There are irregularities in the shape in the lens or the cornea, and when that occurs, this creates an imperfect focusing on the retina. We're off, right? We're off. So here we're like trying to focus. We see at the top this is normal. Down here we see we have light focused here, we have light focused here, right? They're off of the point we want them on. And so because of that, that creates a blurring. This is what we call astigmatism, right? Some people may have astigmatism that gets corrected with glasses. I'm one of those people. And so it's really apparent when it comes to astigmatism at night, because things like street lights, right, stoplights, things of that nature, rather than looking like a single light source, just kind of with a glow, it looks like a starburst, right? Like it's stretching and being pulled in all different directions. It's extra blurred because there are multiple points that light is being focused on in the retina, and it's not on that single point we need. Another visual process that helps us with clear, crisp vision is accommodation or visual accommodation. This is the changing of the shape of the lens I was talking about earlier, where the lens is changing shape to focus at different distances. Basically, as light enters in the eye, right, we want to maintain focus on that one single fovea centralis, but light coming in from different distances is going to need to be refracted slightly differently, right? We need to change the how we're refracting light depending on if the light is originating from a far away source or a near source, right? So we always want to focus on the fovea centralis, and so to do that, the lens will change shape to create different bends, right? Different refracting in the light as it comes in. So in here, we're looking for distant vision, right? We're looking in the distance. So if you were looking at something far away, we see that the ciliary muscles relax, which allows the lens to kind of flatten out, kind of like a little pancake. And so the lens thins out, it flattens out, light still reflects on that fovea centralis. But if we shift our vision from something in the distance to something close by, so for example, if I'm looking out the window, right, across the street, my lens is thin and flat like a pancake. And then something pulls my attention to something right in front of me, something on my desk. My lens has to change shape so I can maintain focus. So what will happen is, as I shift my gaze from out the window to something on my desk, the ciliary muscles contract, and that is going to cause the lens to thicken. It kind of fattens, right? It gets nice and fat and rounded out compared to what it was before. And all of this is to maintain focus on the fovea centralis. So it's constantly making, again, this is an autonomic response. This is constantly making little adjustments in the lens's thin or thickness, right? Stretching it out or fattening or rounding it out, doing that so we can always maintain focus on whatever we're looking at regardless of the distance. Another process is visual acuity. This is the sharpness of your vision, so like how clear your vision is. And really that comes down to resolving power, almost like a microscope's resolving power, right? Remember, resolving power, resolution is just your ability to distinguish 2 points as separate points. And so we see the eye chart. There's one I have here on the slide. We'll have some in class as well if we have the chance. But this is a common eye chart. You do this test, you use these charts from 20 feet away, and you're seeing which levels of lines of text, right, of letters that you can read, right? 20/20 is considered normal vision, right? If you can read the line at 20/20, then, in this case, we see it's the very bottom one there. If you can read that line from 20 feet away, you have normal vision. But not everyone has normal vision. Some of us, like myself, fall into abnormal vision, or we need corrective lenses like glasses or contact lenses to bring us to 20/20. So for some people, right, they cannot read that line at 20/20. They may read it at 20/40 or 20/50. And what those numbers mean is like what this person reads at 20 feet, a person with normal vision would be able to read at 40 feet away. So like 20/40, right? The person taking the test with 20/40 vision would be able to read that line, PECFD, at 20 feet away. Someone with normal perfect vision would be able to read that same line, PECFD, at 40 feet away, twice the distance. And so if we're talking about some abnormalities with vision, some of those include myopia and hyperopia, also known as nearsightedness and farsightedness. I, for one, I have myopia. I am nearsighted. Nearsighted means I can see things close by. The thing I have trouble with are things that are in the distance. Hyperopia is the opposite. Someone with hyperopia would have trouble seeing things up close, but would have no problem seeing things in the distance. And if you notice, all it comes down to is eyeball shape. So here is a normal eye, right? We see the focal plane there, and we see it's nice and rounded. When we look at someone who is farsighted with hyperopia, we see that the eyeball has been kind of squished inward. It's shorter than it should be. Whereas someone who's nearsighted, we see that this person, their eyeball is longer than it should be, which is why we have to use corrective lenses to help refract light on the correct spot, right? Kind of change how light enters the eye so that we can focus on the fovea centralis. Something else we do is we adjust our pupils in response to light. This is an autonomic reflex that will basically adjust the pupil size or diameter by using little muscles in the iris. Basically, this is going to help control how much light enters the eye so that we don't have too much light getting in, and also so that when we're in low light situations, we can increase the amount of light that gets in. We're trying basically to regulate how much light enters the eye and trying to keep it relatively consistent so that our vision is consistent as well. There's two different types of muscles we find in the pupil. We have circular muscles and radial muscles. When we constrict the pupil, meaning we make the pupil smaller, we are going to contract the circular muscles. This will reduce the amount of light entering the eye. So under bright circumstances, right, when the light's really bright, we see that this is innervated using the parasympathetic system, the parasympathetic nervous system, when dim light or when bright light is perceived. And then dilation, this is we contract the radial muscles. This is going to increase the amount of light getting into the eye because we are increasing the diameter of the pupil. This is going to be innervated on the sympathetic nervous side, right? And that's something we see, right, when someone gets nervous or scared or excited, right? Their pupils tend to dilate, they tend to open when they experience that. So if we look over here on the picture, we see what happens, right, when dim light is perceived. The sympathetic sympathetic central neuron is stimulated. We send that along, and then we release norepinephrine so that we can contract those muscles in the iris so that we can dilate the pupils. Whereas when bright light is perceived, right, we go along. I don't expect you to memorize this picture exactly, but bright light's perceived. Parasympathetic is releasing that neurotransmitter, and pupils constrict. Now, something else that happens is called convergence. This is where we are basically using our eyeballs together, right? Our eyeballs work together to create the entire picture, and they're moving in a way so that they are both directed towards an object, right? That means the visual field for each eye overlaps, and this is what gives us what's called binocular vision. And binocular vision is responsible for giving us what we call depth perception, basically your ability to see things in 3 dimensions. Another important thing to note is that vision is like the one thing that is neither contralateral nor ipsilateral. Remember, contralateral means that the body part is controlled by the opposite side of the brain, and ipsilateral means they're controlled by the same side. And I know you're thinking, wait, what? If it's not opposites and it's not the same, what the heck is it? Well, it's a mix. So if we look here in the picture, we see some information in red, right? This information red hitting the sides of the eyes, and we see that those 2 red lines, right, from here, we're going to be ipsilateral, but on this side in the right eye, we're contralateral for that. And on the other side, we see the blue arrow, right, and the blue pathway, right, we see on the left side is contralateral, but on the right side is ipsilateral. So each eye, right, has an ipsilateral side and a contralateral side. So it's neither, right? You could say it's neither, it's both, right? Whatever. But it doesn't fit the contralateral or ipsilateral definitions explicitly, right? We see a blend in that both sides of the brain are receiving information from each eye to give us the entire picture. Then we get to photoreception, right? For us to be able to see, for vision to happen, it requires us to absorb light by photopigments, right? Photopigments are special pigments that can absorb light in the form of photons. And in this case, right, when we're looking at human eyes, we're looking at rods and cones. Photopigments are going to be found inside these rods and cones. They are proteins found in there that have the ability to absorb photons. And when they are hit with light of certain wavelengths, they actually will physically change shape. They go through a structural change. When we look at rods, we have one photopigment, and that's called rhodopsin. And in cones, we have 3 different kinds. One for red, one for blue, and one for green. Right? Basically one for each different type of cone. And photopigments, when they respond to light, it's actually very cyclical. And so we're going to kind of walk through this, right, kind of, sort of, and talk about really just focusing on the rhodopsin, talking about what happens in the rods. So in a rod, we have this rhodopsin, and when it is dark out, right, when we are not exposing that rod to light, the retinal, right, inside the opsin is what we call cisretinal. And this has a little bend in it. So if we look over here, here's our cisretinal. And opsin, when they are found together, we call it rhodopsin, right? So rhodopsin, right, together is retinol and opsin. And here, if we look at the retinol, we see there's a little bend in it, right? Then we expose the rod to light. When we do that, the retinol will absorb the photon and it's going to go from this little bend to straight, right? It straightens out to what's called transretinol. When that happens, right, a G protein gets activated. This causes depolarization and causes an action potential so that we can understand like that signal as we saw something. This also leads to what's called bleaching, which is right here. So that transretinol actually, because of its changed shape, will actually separate from the opsin, and now it's colorless, right? This is why we call it bleaching, because we have lost our color. Kind of like if you put bleach on a shirt, that shirt color would disappear. Now, when we use an enzyme and we use ATP, we can convert from transretinol back to cisretinol. And then because we're back to cisretinol, that little bend is back in the molecule, we can form back with opsin, and we are now rhodopsin again. So we're basically seeing this back and forth of retinol with opsin, right? Retinol getting bent. When it gets bent, it leaves the opsin. To put the retinol back in the opsin, we have to use an enzyme and ATP to go back to normal and bind with opsin again. We reform the photopigment, we've regenerated, now we can be used again, right? This is how we detect light, right? We're just going through this over and over and over and over again. Now, cones are less sensitive to light, right? So they require more light in order to function. But it helps us with color vision and it gives us crisper, clearer detail. We have those three different cones. We have green cones, red cones, and blue cones. They have their own opsins called photopsins or photoopsins, as well as their own retinols. We're not going to get in the details of how each one works, but just know they have different photopsins and that they depend, which ones they respond to depends on the wavelength of light and the intensity. So if we look here in the picture, we see what wavelengths of light, kind of trip that particular cone. So blue cones are going to be activated, or they're going to be triggered by ones in these shorter wavelengths, like 420 nanometers, whereas green is like in the mid 500s, red's even further along. So we have like short, medium, and long wavelengths. Blue tends to be triggered by short, green by medium, red by long wavelengths. And of course, we can't get out of this one without talking about what can go wrong. Here's just a few different things like cataracts. Cataracts is a cloudiness of the lens. Sometimes cataracts can be small, but it can also be very large, so much so that someone's vision is completely obscured. Depending on the cataract and the size, a person may or may not need treatment. In many cases, surgery can correct it, and it's something that is done, we've been able to do for a long time. It's something that is really successful, right? It's a really successful treatment for cataracts. You see it happen all over the world. In fact, like some places will take donations to do this in all parts of the world. So they'll just go through and do all sorts of cataract surgeries for people who absolutely need them, who otherwise would not have medical treatment. Glaucoma, this is when the intraocular pressure is very high. Basically, the aqueous humor in the eyeball has built up. And so this actually puts pressure on the neurons and destroys them, leading to blindness. Presbyopia, this is basically when, as you get older, visual accommodation starts to lessen. The lens gets harder. And as the lens hardens, it can't flatten or squish anymore, and so focusing on near to far becomes more difficult. This is one reason why you see people need like reading glasses as they get older, right? Even though they may have never needed glasses as a child or even a young adult, as they get to middle age and older, they seem to have trouble, right, with their vision because they can't make that adjustment from focusing on something near to far. And so usually we see older people needing things like reading glasses. And then lastly is color blindness. This is where a person's color vision is distorted. It is lessened. It doesn't mean a person sees in black and white. There's actually different forms of color blindness, but all of them lead to a muted sense of color because they are either missing or have a deficient version of one of those three cones. Remember, we have red, green, and blue, and so one of those cones, right? There's a genetic problem that leads to one of those three types of cones either not being there at all or not functioning properly. This is more common in males. We do see that as sex-linked, and so it's actually, compared to other conditions, relatively common in males, but is much more rare in females, right, because of it being linked to the X chromosome.